Okay, so I think I'm going to start. So, good afternoon, and my name is Dan Sliva, and I will be talking what we are doing in the lab and how to identify effective cannabinoids. Because the major problem in this industry is the real effectiveness. So the medical benefits of CBD, mainly we can focus on CBD, are well known, and they are used. CBD is used against different diseases and medical conditions. We are not supposed to say diseases because if you are talking about diseases, this is in the focus of FDA. So, but still, people are buying CBD and cannabinoids because they want to help themselves. And what is CBD? These are different forms of CBD which are on the market. The first one, which actually was the most popular, was the CBD isolate, and that is the purest form of CBD, and it means the other cannabinoids and other compounds in cannabis were totally removed. Usually, this is recommended for people who are specifically taking high doses of CBD. Some people can be sensitive to THC or other cannabinoids, so again, only CBD. And other people, they are going through the drug screening, and in this case, you don't want to have any THC in your blood. Some people may prefer different flavors or no flavors. And some people actually, they are living in states that there are strict THC laws, and that is actually directly here in Indiana, because we are limited to 0.3% of THC. Plus, in addition, sometimes the first-time users, they are scared to use other cannabinoids, so they are focusing on CBD. So that is CBD isolate. Another form, really popular, it's full-spectrum CBD. And CBD has been extracted, and it has been extracted with other cannabinoids. And that is the full benefit of the cannabis plant. It's called entourage effect. And this one actually may have psychoactive, sedative, or other innovative side effects. It may show up on drug tests because you have certain levels of THC. And in this case, THC may cause legal complications, because the point is that still you can have you have full spectrum, you have 0.3% of THC, but you are dosing and dosing and dosing, so it means your level of THC is accumulating. After that, you are going through drug test, and you are not going to pass it because you are B positive. In some cases, it's true. In other cases, not, because some other cannabinoids actually they can cause false positive effect. So that is some problem with the full spectrum. Otherwise, people living in states where cannabis is legal, it is absolutely no problems. So for that reason, it is such a called broad spectrum CBD or CBD-free THC. So it was prepared by practically the same process. You have all the compounds in cannabis, but THC was removed. And again, there is a little bit limited production because to remove THC from the full spectrum, it's a little bit technically complicated. So these are three major CBD products. So, and now, if you are talking about CBD effectiveness, and this is really busy slide, and I know, so for not scientists, it's probably too much, but please focus on the right side on this anti-inflammatory effects. And I know I'm gonna drop from this one because it's really, okay, if you wanna take a picture, it's really complicated, but it means CBD, works not only through CB receptors. It works through different receptors, through different signaling pathways in cells. So it means CBD has different biological effects. So I know it's complicated, but we can move with it. Okay, so for that reason, actually we are, and this is the simplified version, we are in the laboratory. We have specific assays that we can determine anti-inflammatory effectiveness, because you can see everybody is claiming that their products are effective, they are anti-inflammatory, whatever. However, if you are not going to test it, you really don't know. So in this case, I'm going to jump down. We can induce inflammation in cells, and if we add CBD, we can actually block this inflammation, so the cells are also affected. However, if there is no CBD, the inflammation has real negative effect on cells. So we can quantify this effect if CBD is really effective. And why is that important? Okay, back to the lab. And these are actually data from our laboratory. And this is really interesting because this is the CBD isolate. So this one supposed to be really pure CBD. And we purchased this CBD isolate from different producers or sellers. And what is interesting is that all the time we have a certificate of analysis. And on the certificate of analysis, it was written all the time that is something like more than 90%. And in one case, it was more than 100%, which is nonsense. And you know, this is certificate of analysis. So we run anti-inflammatory effectiveness on the assay, as I showed you before. So it means in cells we can stimulate inflammation and we can block it. And you can see there is different effectiveness. It was really strange because all of them supposed to have the same amount of pure CBD, close to 100%. So we analyzed by using HPLC, we analyzed the amount of CBD, and you can see there is not really not the same amount. So it's really going down. Plus, in addition to that one, you would expect the highest level of anti-inflammatory activity in CBD1, which is 95%, according to our analysis, which is fine. But CBD4, when we found 86%, is actually higher anti-inflammatory activity. In addition to that one, CBD2, 94%, and it's only 39% effectiveness. The same like for 88% CBD and 56%. So the point is that what is really necessary to, in addition to certificate of analysis, you have to really know the biological activity to be sure that your CBD is effective. So in addition to CBD, we also analyzed different cannabinoids. So in this case, we analyzed THC, Delta 8, one CBG, another CBG, and CBN. And what is interesting, you can see THC only 17% effectiveness. However, this THC is supposed to have, according to their certificate, something like 100% of THC, which was not true. Again, we analyzed in the lab the amount, and the real amount of THC is this one, 39%. However, there is total amount of cannabinoids is 80%, because in this case, it was 39% of THC and something like 39.5% THCA. THCA is precursor of THC. THC is formed from THCA by the process of decarboxylation. However, if you are looking for the strict effect of THC, you would expect that it's going to work. And you can see only 17%. In addition to that one, Delta 8, which is really popular now, 41%, and CBG1, one CBG, another CBG, really good, relatively good activity, CBN something like 70%. So that is why I'm repeating all the time how it's really crucial to analyze biological effectiveness. Because again, you can see something on COA, you can analyze it yourself in your lab by HPLC and you say, okay, this is fine. I have high amount of cannabinoids, but the real effect, it's not, doesn't correspond to amount of cannabinoids because you don't know what else is actually in this extract or you know, this product. So we analyze the effectiveness of full spectrum, and you can see there is some correlation that with the highest level, you can see 82% anti-inflammatory effectiveness, which corresponds to 80% of CBD. But you can see there is, actually this is full spectrum, so you can see 4.3% THC. This one, by the way, again, in Indiana, it's still illegal because it's 11 times more than legal limit 0.3. And you can see the other full spectrums, they have also different effectiveness. And you can see different amount of CBD and THC in these extracts. So the major point is that really COA is important, but how it's true. And we analyzed another full spectrums, and again, all these full spectrums on their COAs, it was something like between 80 or 90% of cannabinoids. However, as you can see in the last one, sample 9, full spectrum 9, there was only 30% of CBD. However, you know, you can see 32% of the effectiveness, which, if you compare with other ones, is not so bad, because if you compare it with the sample full spectrum 5, you have practically 67% of CBD and 36% of effectiveness. You are going down by half by, in the last sample, first full spectrum 9, you are going to 30%, but you have only half amount of cannabinoids. So that is, again, the confirmation or the necessity to really analyze and validate everything. So, okay. And this is the broad spectrum. So it means full spectrum stripped of THC. And again, this is practically the same amount of CBD in this full spectrum, and you know, the same amount of total cannabinoids. However, you can see 50% tree efficiency or 75%. So this is, it really doesn't correspond what is inside. And again, before, when we run the experiments, we received, you know, the COAs, and you know, you trust the COAs. Actually, I don't trust it, because you know, in some cases, and I guess I mentioned it last time in the other discussion, that you know, I had certificate of analysis and that there was something like 102% of CBD, which is nonsense. So I was surprised, you know, if these are real, for real, these guys in this company, because you know, common sense, you know, why you are selling something and declaring that is something which is impossible. So anyway, so this is the broad spectrum. And okay, so I think you heard a lot about this entourage effect, and entourage effect is great, but in my opinion, in our opinion, we are looking for the best effect. So you have entourage effect is the full benefit of the whole cannabis plant or on the whole extract. So how you can have the best effect? So it means something better than entourage effect. So we looked for the combination of different compounds, effective compounds in cannabis, and we were actually looking for the synergistic effect. And you have additive effect, mathematically additive effect, it means 1 + 1 = 2. If you are looking for synergistic effect, you are looking for something like mathematically 1 + 1 is more than 2. And I know it seems to be crazy, but you know, actually it is how the biology works. And okay, so we were testing it, and now, so again, for anti-inflammatory effectiveness, we can see nice effect with CBD, and we found actually nice effects, anti-inflammatory effects with terpenes. But when we put CBD and terpenes together, you can actually see the practically better effect. And no CBD, no terpenes, the cell or cells are surviving, but they are suffering from inflammation. And okay, so it is a little bit busy. So this isobornol, which is one of the terpenes. It was alpha terpineol, and you can see only 18% and 20%. And to explain, we can actually, if we increase the dose, it's gonna go up, okay? And but we were looking for the really small effect. And there was the really small effect, this isobornol and terpineol, this full spectrum, something like 50% effect. But when we put together full spectrum and isobornol, you can see 89% anti-inflammatory effectiveness. So it means 1 plus 1 is more than 2. Or in other words, 18 plus 51 is less than 89. The same is actually for terpineol, 20% plus 51. Together it should be, if it's additive effect, it would be only 71. However, it's 95%. So that is how we are trying to create, really the best effect, the best combination of different compounds. So, okay. In addition to terpenes, we were looking for some other things which are not directly linked to cannabinoids, and these are really popular essential oils, because some of the essential oils, they have really great activities, like other natural compounds. So we run the same scenario in the lab, only CBD, essential oils, CBD plus essential oils, or CBD without essential oils and without CBD. And these are the data. So we took the CBD and we went to something like close to 50% of effectiveness of CBD. And when we tested the lemon oil and sage oil, you can see this is some small effectiveness. And again, it's created actually artificially, because we can do the overkill. We can put more amount of lemon oil and we are going to have 100% effectiveness. Or we can do the same with the sage oil. But the point is to go down with these oils and with these CBD and put them together, because by that, actually you can save money if you are creating your product, because you can really go down, with the amount of active compound. However, you can see really nice biological effect or anti-inflammatory effectiveness. So that is what, and actually we are also using that one for some other natural compounds, because it's possible to combine CBD with turmeric, with green tea extracts, and with other products. In addition to anti-inflammatory activity, actually we measured, and I'm not going to show it here, antioxidant activity, because this is really important, to have more activities, because some compounds, they are active in one pathway, other in different pathway. You are going to put them together, and you have the best effect. And okay, so as I mentioned, entourage effect is great, but we are looking for the best effect, to have really something which is strong. And okay, there was a lot of talks. I added this one in the last minute, that why we are testing this effectiveness, because these are 3 different brands. One is CBD, some CBD product. I'm not going to put what was the brand, in oil, and you can see 32% effectiveness. The same amount of CBD in the product, in the hemp seed oil, suddenly you can see only 4%. However, CBD in nanoparticles in the product C, you can see 100% effectiveness. So it really depends on the formulation. So the question is why, And that is for that reason, because effectiveness depends on CBD absorption, which is directly linked to bioavailability. So normally CBD has between 6 to 8% of absorption. I think you heard it before, from in some other talks, because it's lipophilic. It means it's soluble in oil or in lipids, but not in water. And that is the problem that somebody can give you, okay, this is the great CBD, and there is only 8% or 6 to 8%. Some products are actually, the people are using oil to dissolve with oil. By that you can enhance the absorption by threefold. It means you can go to some 24%, but still it's not 100%. So the best way is actually to have CBD in specific nanoparticles that you have practically 100% absorption. So in other words, again, in nanoparticles you can use less CBD and you have better effect. So that is, and I think that, yeah, so it was the last one. So if you have some questions or this for discussion. Yeah.
What other oils would you say, you do equate like MCT oil to being an example of oil there? Is that different from, say, the other rapeseed or the...
Yes, I would say the oils generally, they are you can dissolve CBD in different oils. And there is no, I would say, better oil, or it's the choice. Because, and again, there is one thing, you can have some flowerings, because CBD, some people, they don't like, the smell or taste of CBD in oil, because especially CBD in hemp oil, you can really taste it. It's different, so you can add some other flavoring or to mask it. So it depends. But I would say if you are asking actually for what is going to be the best oil, I think the best oil would be something what we tested or what we can test for the activity. Because some hemp oil itself, there is no activity. However, as I showed you before, it was with this lemon and sage oil, you can actually see some anti-inflammatory activity. So that would be probably the better choice. But in this case, you are going to have some specific taste or smell. So again, so it depends. So the people, what they prefer. But in my opinion, the best is, number one is, to have something what is effective. And after that, you can add something. However, you add something what is necessary to test it again, because that is what we found, that some kind of compounds which are used for only, some kind of like fillers. Yeah, fillers, this is a good term. Fillers actually can really kill the activity. That is what we found. So that is... Okay, this one?
Is it just a specific brand that made it lower, or the hemp seed oil that made it lower?
No, it was brand where actually CBD was only dissolved in hemp oil.
So not simply the brand.
Yeah. Yeah, I don't want to put the names of the brand because...
Feedback off the other discussions, are there certain terpenes that, and things that you look at in additives that really stand out effectiveness, like those, couple you mentioned for inflammation?
Yes, we specifically, because when we are, creating something, or usually the clients are coming to us and they are asking if we can, put together and test, some product or formulation. So the major question is, what is your target disease or conditions? And after that, we are going through specific medical literature to find what is published and what is known. And based on that one, actually, we can coming up with some ideas what we can test. And it was especially in this case, it was anti-inflammatory activity. And so we were searching different oils in hemp, and it was also they were interested in some specific scent. So, and we found that actually in this case the lemon oil and sage oil, and we ran also some other with jojoba oil. It has some activity, plus it was pleasant smell on actually with the clients. Whatever. And it actually, yeah. Oh, sorry, I jumped. Okay. Yeah, this one. Yeah.
And so with this, for an example, if you have a product, let's just call it like 35 milligrams of CBD,
Mm-hmm.
what sort of dose with that CBD are you looking at for those couple terpenes, rather than just being detected, but like actually?
Oh, okay. What we are usually trying to find. Okay, I'm gonna be specific with this one. Okay? So, what we are looking in the lab, we are looking number one, what is IC50. IC50 it means it's 50% inhibitory effect. So it means in this full spectrum we found a concentration which can inhibit 50% of inflammation. Okay, and after we were testing different terpenes, and we found actually that we can go down with the amount of isobornol and terpenol, and still we are going to have the really great effect, because you can mathematically you can something to write on the paper and to test the hypothesis, but you have to confirm in the lab. And actually we confirm that actually we can use this small amount, which has only 80%, but when we are going to mix it together, we are going practically to 90%. I don't know if I'm sure.
I guess my question would be like, say to get that 18%, what's like amount of, say, milligrams of that terpene is being added to get to those levels?
Okay.
So volume and a dose.
Yeah, it was something like this. This terpenes, it was in the amount of less than 0.1% of terpenes. Yeah. So they're definitely not milligram levels, but microgram levels.
Right. So it's really because of the levels of detection. If you are detected,
Mm-hmm.
is that likely an adequate amount? You wouldn't necessarily see a detection and say it's not enough per se.
Yes, this is the reason why it's necessary to test it, to optimize the conditions and to find what is the best ratio, because it depends from compound to compound. Plus, in addition to that one, and it was especially in this case, you can find this great ratio which is really effective. But now, this is CBD or full spectrum, and you can have the best effect if it's going to go to nanoparticles. Because one thing is if you are optimizing conditions in the lab, and another thing if you want to optimize it for the final product.
When we make a formulation, we can test obviously the levels that are in there, but how do you actually test effectiveness in that?
What were you saying, please?
Like we can test it and get, you have this percentage of CBD or this milligram. I mean, that's the data we get from testing in a lab, but this is effectiveness, like anti-inflammatory effectiveness. So how do you actually test that?
You mean like in animal studies, or we can run some animal studies also, anti-inflammatory, and in human studies we were not able to do any clinical trials, but we were able to create some products actually, and based on the response from the clients, actually returning clients, it was effective. Yeah. But I would say number one, before we are gonna start to sell something or promote something, we should know if it's really effective in cells, because if it's not effective in cells, this is the first step. It's not gonna be effective in human body.
Just curious, have you done animal studies?
They've done animal studies, but not with CBD, but with some other stuff, because, okay, it's complicated. Okay, these animals, if you are looking for inflammation, this is called endotoxemia. Okay, so it means you have mice, and you are gonna hit these mice with endotoxin. And it means they are gonna have huge inflammation. They are gonna have septic shock. And they are gonna die from the septic shock in 1 or 2 days. This is really harsh. And you are gonna test your product of interest or your compound, and you are trying, to avoid or to postpone the tests. This is not the best way. What we actually develop these animals, mild model of endotoxemia, because in this case we can induce inflammation but not septic shock, and still we can measure some compound there anti-inflammatory. So why? This is really important because all of us have some certain level of inflammation, which is sometimes impossible to do. For example, if you're doing some sport, you are a runner. Okay, this is what I have because I'm crazy now. So if you're a runner, you are inducing inflammation, you are inducing pain of this inflammation. So you want to stop this inflammation to have some pain relief. So in this case, you can actually work testing your stuff on your side. So I would say this is something like, okay, why I'm saying that is actually what I was doing, my whole life, in academia, to look for some natural compounds used for prevention. Because in some cases, you have started inflammation for, let's say, 10, 20 years. This inflammation eventually it's gonna develop in cancer. It's gonna develop in atherosclerosis because it was undetectable at the beginning, because it was really small amount of inflammation. We don't know it because undetectable even at an exercise. In 20 years, because it was in my previous life, I was looking, I was looking how to use natural compounds for prevention in oncology. So, and actually the same actually applies to cannabis and to CBD, CBG cannabis, terpenes and these cannabis. But again, this will be repeating again and again. Number one, what is really important to test the real effectiveness or efficacy of biological activity.
Can you speak on the perceived effectiveness for CBD specifically?
What do you mean?
Like what health benefits it's perceived to have, separate or that may be different from CBD itself?
Is CBG different from CBD?
Okay, got it. Okay, so CBG is also anti-inflammatory, and this is the beginning. It was the first slide. It was the beginning. It was this really complicated one. Okay, because this is CBD. CBD, everybody is talking about CB1 and CBD. Okay. CBD doesn't activate CB1 and CBD receptors. This is the first misconception and first BS what the people are using all the time. It's total nonsense. Because, CBD is allosteric negative regulator, focus on negative. Okay, I know this is pharmacology. But it means that actually CBD binds, it's able to bind to these receptors to block their activity, their function. So in this case, CBD is used as an antidote for THC, effect. If somebody is poisoned or overdosed with THC, the best way is use against this THC to use CBD to block this effect, to go to CBG. And it means CBD, it goes through different pathways or receptors in the human body. That is what is known for now. CBG, it's, this is the beginning of the studies with CBG. It's not known exactly how it works. In some cases, maybe there's going to be overlap with CBD. In other cases, it's going to go through different pathways. So the reason is because these are cannabinoids. It was forbidden or prohibited in academia. When I was in academia, you are not allowed to touch CBD, which is not psychoactive, or CBG. Cannabinoids, nothing. No. So, and that is the reason, that is what I repeat in this catch-22 actually. These are not enough research. Why to move forward with cannabinoids? Why it's not enough research? Because it was prohibited. So, yeah. So to answer your question, CBG is anti-inflammatory. We don't know the exact mechanism with other stuff. So, and I think that is so important to have the biological assays to really show if you can see some activity, some effectiveness.
But I've always heard about CBG recently, that it's more about the GI tract.
Uh-huh.
Have you heard of anything?
About GI tract,
Yeah.
some people doing some adverse effects or some positive effects.
Yeah.
Probably I would guess that if it's going to overlap this CBD, because CBD in some cases it has anti-obesity effects, other effects, calming effects. So I don't know. To be honest, I don't know. But it's possible that there definitely is that. But for example, some people when they are using cannabinoids, they can feel some discomfort by the oral.
Is that what you heard about GI tract, or is that what you read?
That's what I've heard. I mean, I've read it mostly through other brands marketing, right? So I haven't read studies on it.
Right, that's probably the most important thing to see a scientific study on it. Does it have an effect on the GI?
And what is like other people say, well, take the CBD products for anxiety and then take this for more like gut health.
Yeah.
And that would be great, but
I think I get what you're saying. There's no proven studies on the CBG for effectiveness of the GI, or there is on the CBD. Because it was prohibited for all those years. So,
And because whatever came from cannabis, cannabis is hemp or marijuana, whatever came from cannabis, no way in academia, no. So,
You have a recommendation or something you've seen on average when people are looking for CBG as to what a average dose would be, whereas let's say 35 milligrams again is fairly common for CBD.
what actually what we found, I didn't show you here, what we found in the lab, again, that when we put together CBD and CBG, we have definitely better effects.
the combine the two.
Hmm?
You combine the two?
Yes. Yeah. But again, everything depends on the price, because the price of CBG is really higher than CBD now. But there is different aspect, because CBD, I wouldn't say it's legal or illegal from the FDA. FDA is silent now, because the CBG is going to be a different story, and let me explain why. Because CBD is in Epidiolex, which is a drug. If you have something which was approved or tested for the drug, it's prohibited by FDA to be used in dietary supplements. However, gyms are viral, so FDA is not able to stop this way. But it's important to regulate this way.
In your research, when you found that CBD and CBG were better together, what type of ratio did you have of the CBD to the CBG?
And again, it depends on the source. Because as I showed you in one of the slides, that Okay. So, as you can see, that it was practically the same effect of CBG, but you can see now one was 92%, another was 83%. But definitely the effect is there. So again, it depends what is your source of CBG and what is your source of CBD, because after that you know it, or we can find it and we can put it together and to find the optimal ratio for the best activity. Yeah. Okay, you mean? Yeah, but and what I'm repeating, I had COAs which came actually with these pure CBD isolate, and it was something like close to 100% CBD, which, according to our analysis, it's really there. But it should be really pure isolate. So how is that possible? It's something which should be really, really pure, only such a huge difference.
I guess could we have an example? Say you had two sources, a CBG isolate that's 95%, a CBG isolate that's 95%. If you wanted to work effectively together, assuming they're the same levels, what would that ratio maybe look like?
Again, it depends. It depends because still we can run CBG isolate and we can see high, let's say this 95% of CBG, but because this analysis is only focused on cannabinoids, we don't know what else is there. So, and there should be maybe something which can suppress or inhibit activity. So to answer it, the best way is to test it. But definitely you have to know something to start with. You have to know the amount. It is there and after that. And I can tell you from the same supplier of CBD, the same certificate, probably recycled, totally different. So that's the problem. So. Okay, so thank you. There is no questions? No. Thank you.